Okay, at this stage right here, you guys, the pyruvates are already made. Glycolysis is over, and a decision has to be made. What happens to the pyruvate? Does it go into the mitochondria, or does it become fermented? Now, there's a good question that a student might ask me and say, why? Why do you have to get rid of the pyruvate? Remember, at the end of chapter 8, we said, the product of a reaction, if it builds up in concentration, it will negatively feedback and inhibit the enzyme. Do you remember all that? I'll say it really slow now, all right? Because I said it really fast. Negative feedback inhibition says the product of a reaction, if it accumulates, it will negatively feedback inhibit the enzyme that made it. That's regulation. You don't want this to happen. You don't want pyruvate to turn off glycolysis. Because at times when you don't have oxygen, at times when your cells do not have oxygen, they're actually surviving off of these ATPs until the oxygen comes back. If you turn this off, that cell is gonna die like that. So you must get rid of the pyruvate. All right? There's another problem. You also have to have empty trucks all the time. Say, look at it. This is a tough one. A lot of students miss this one. There's a dilemma. There are two problems for the cell. Number one, I have to get rid of the pyruvate. Number two, I have to get rid of pyruvate to keep glycolysis moving. Number two, to keep glycolysis moving, I have to always have empty trucks. If you don't have NAD+, glycolysis will shut down. So glycolysis can shut down in one of two ways. If you run out of this, or if you don't get rid of this. So your cell has two problems. Guess what it comes up with? One solution. One solution to fix both concerns, to address both concerns. I'll give you an example of what would happen, what is similar to not having empty trucks. Not having empty trucks is similar to this. You're moving to a new house on Sunday. You call U-Haul. Hey, I want to rent a truck. U-Haul says, we don't have any empty trucks. That means you're not moving on Sunday. That's what it means. Hey, I don't have any empty trucks. That means you cannot do glycolysis. That means that cell is dead. So your cell decides, you know what? I have to come up with a solution. Here's the solution. To get rid of pyruvate, I need electrons. Oh, I just loaded the electrons on the truck. So I'll unload them. Watch this. I just loaded the electrons on the truck thinking that I'm moving out. I just loaded the furniture on the truck thinking my new house is ready. But surprise, my real estate agent called me and said, your new house is not ready. So I have to unload the furniture. Once you unload the electrons to give it to pyruvate, that means you have an empty truck again. Am I right? When you unload the furniture off your truck, that means you have an empty truck. It's clever. To get rid of pyruvate, I need electrons. I'll get them from NADH. And when I do that, I empty it. So I will always be getting rid of pyruvate, and I will always have the empty trucks. Problem solved. Actually, both problems solved with one solution. So let's talk about the fermentation part. Without oxygen, both pyruvates will be disposed of because I need glycolysis to keep going by fermentation. Now, the end product of fermentation depends on which cell you're talking about. Are you talking about an animal cell like you and I? The end product will be lactic acid. Are you talking about yeast? The end product will be ethanol. And thank God for yeast. Because without ethanol, You can't make wine, and you can't make my Heineken. I love Heineken, right? And then bacteria. The interesting thing about bacteria is they're variable. Some bacteria can make lactic acid, some bacteria can make ethanol, some bacteria can make a whole mixture of things. We're not going to worry about them. We're just going to say variable. If you want to learn more about this one, that's microbiology. So, at the end of fermentation, what do we have now? Well, we don't have the pyruvates anymore because they were fermented. If you're an animal cell, you now have lactic acid, not pyruvate. If you're yeast, you now have ethanol, not pyruvate, and so forth. But energy-wise, you have 2 ATPs. Remember, the net 2 ATPs made by glycolysis? Here they are. I just told you that the loaded trucks made during glycolysis, they get unloaded. So these 2 delivery trucks, unfortunately, they're not going to be delivering anything because they get unloaded. So the only thing you got to get out of fermentation is 2 ATPs. That's it. The fun begins when you have oxygen. And like you're doing right now, you're breathing. You're not doing fermentation. You're doing this one. You're doing the full movie one. So the fun begins when you have oxygen, and here's what happens. Instead of the pyruvates fermenting, they're instead going to enter the mitochondria, because we're dealing with eukaryotic cells here today. So both pyruvates will enter the mitochondria, one at a time. How many carbon is pyruvate? It says 3C, 3 carbons. So watch the show up here, please. The pyruvates, here it is, 3 carbons. How many do I have? I have 2 of them. No oxygen, fermentation. Yes, oxygen, I will go towards the mitochondria and I will enter the mitochondria. And as I'm entering, watch this, as the pyruvate is entering the mitochondria, watch, it loses a carbon and becomes acetyl-CoA, 2 carbons. It's a different molecule now. As the second pyruvate comes in, it also loses a carbon and becomes acetyl-CoA, 2 carbons. And that's what this one is showing. The pyruvate enters the mitochondria. Each pyruvate loses a carbon. We have 2 pyruvates. That's why you have 2 carbons lost in the form of CO2. And that's what you're breathing out right now. And then acetyl-CoA is the leftover part. Look at this. This is called pyruvate oxidation. The breakdown of pyruvate into acetyl-CoA. Interestingly, you also make 2 loaded trucks here too. Now, some students by mistake, they think that these 2 loaded trucks are the same as the 2 loaded trucks from glycolysis. No, they're not. They're 2 different ones. You actually have 4 of them now. 2 from glycolysis and 2 from pyruvate oxidation. So let's do the tally for this one. At the end of pyruvate oxidation, this is your tally. You have 2 ATPs made from glycolysis. You have 2 NADHs also made from glycolysis. By the way, these 2 NADHs will enter the mitochondria too. In the presence of oxygen, these NADHs will enter the mitochondria just like the pyruvates are entering the mitochondria. You will have 2 CO2s that you will lose because they're toxic to you. They're just waste products of cellular respiration. So these 2 CO2s are produced during pyruvate oxidation. Very good question. Did we lose carbon in glycolysis? No. When was the first carbon lost? Pyruvate oxidation. Again, you can tell if you know the flow chart, the entire flow chart. You got this exam, the exam 3, the cellular respiration part. You'll be like, I got it. Yeah, but you have to remember it. Understanding it is one thing, of course. It starts with understanding, but you have to go to retention mode. And I'm telling you, it starts tonight. I know you have an exam on Monday, but you must factor in a couple of hours in the next couple of days or so to start remembering this. You go ahead and wait till the night before the exam. You will never remember this the night before the exam. No way. To start remembering this information, I'm telling you, take my word for it, it starts tonight. I know you have an exam on Monday, but you also have this monster to deal with. Factor in some time, please. Start studying cellular respiration and start getting into the retention mode. You got to remember it. How many of this and how many of that and how and where and what's happening and all that stuff. You got to remember it. Because a lot of students come to me after the exam and says, I couldn't remember it. And their grade, of course, would be really bad because if you can't remember it, forget it. It's not going to work. All right, 2 NADHs from pyruvate oxidation right here. And then the 2 acetyl-CoAs. So that's the accounting at the end of pyruvate oxidation. Okay, the following step is the citric acid cycle. So this is the overall view. Again, the 2 pyruvates enter the mitochondria. They become 2 acetyl-CoAs because they lose the carbon dioxide. In the process, you load the trucks. The 2 NADHs that are made from glycolysis, they also enter the mitochondrial matrix. So all the NADHs are gathering in the mitochondrial matrix. It's like they have a meeting, and they're all going to the meeting. And the meeting is in the mitochondrial matrix. So now you have 4 NADHs, and all 4 of them are in the mitochondrial matrix. Remember, the mitochondria from your organelles chapter is actually made up of 2 membranes, an outer membrane and a folded inner membrane. That means there's a space between them. It's called the intermembrane space. And I did not draw this membrane like this just to be cute. It's on purpose. If I flatten this membrane, you're in trouble. You need this space, and you'll see why later, Monday, oxidative phosphorylation. Okay, so now we're going to start the citric acid cycle. I used to teach it like this. I give them the circle, and I say acetyl-CoA, and this is what you get out of it, and just call it the citric acid cycle, and say, it's okay if you don't know the detail, it's okay. And then the student comes to me to ask me a question about it. When? I'm not kidding you. During the exam. The student would come to me and say, what is that? They're asking me, what is this? On the exam. I'm like, what? I can't answer that question, first of all. And now you come and ask me what this is? Yeah, because I was trying to be nice to them by trying to keep it simple. So I decided, that was like 3 years ago I decided this. I've been doing this for 3 years now. I decided I'm going to be nice to you and give you the whole thing. Because I don't want you coming on the exam and asking me, what is this? Because I know, I get this feeling that, oh my God, if you don't know what this is, you probably don't know what any of it is. And I can't afford that. So I'm going to teach you the whole thing. Now, in biochemistry, they talk about the enzymes involved, the molecular structures of these molecules. I'm not doing that. So, but I'm going to get a little bit more detail than just the circle part, because I need you to know it. Now, I'll tell you something. Once I started doing this, I never got that question on the exam anymore, because they know it. So here is the event. And we'll finish with the citric acid cycle, and we'll stop there. The event starts with this one here, called oxaloacetate. It is not called whatchamacallit. It is not called the whatever. No. What is it called? Oxaloacetate. You are held accountable for everything you say, do, and write. You want a raise, you want a promotion, you have to speak like you deserve it. You have to act like you deserve it. And it's simply pronouncing the words properly shows your boss that, oh, you really know how to do stuff here. It's pronounced oxaloacetate. And if you think that's a tough one, wait until you see the other one. So oxaloacetate is a 4-carbon molecule. It will combine with acetyl-CoA, a 2-carbon molecule. What did I just say? It will combine with it. I did not say that acetyl-CoA will become oxaloacetate. I didn't say that. I said this. Oxaloacetate, a 4-carbon molecule, will combine with acetyl-CoA and make citrate, a 6-carbon molecule. That's what I said. And if you look at the arrows here, that's what this means. 4 plus 2 makes 6, citrate. Then what happens? Citrate isomerizes and becomes isocitrate. You don't lose the carbon. It just changes form, that's all. Isolate. Then what happens? Remember, we're doing the movie. Then this is the one. Alpha-ketoglutarate. Say that 10 times, right? Isocitrate becomes alpha-ketoglutarate after it loses a carbon. Look at this. The 6-carbon isocitrate becomes the 5-carbon alpha-ketoglutarate. That means it lost a carbon. And every time you lose a carbon, you load a truck. I will show you how to remember it. I'll give you some suggestions, okay? But once it all fills out, I'll show you. Alpha-ketoglutarate will lose a carbon and become the 4-carbon succinyl-CoA. Alpha-ketoglutarate will become the 4-carbon succinyl-CoA. Again, every time I lose a carbon, I make a truck. Succinyl-CoA will become succinate. In between these two, you're going to make an ATP by substrate level phosphorylation. Succinate will become fumarate. Oh, look at this, a Penske truck. So between succinate and fumarate, you're making the other truck, FADH2. This is the only place you make FADH2. Ooh, I think that's a question on the exam. The only place you make FADH2 is in the citric acid cycle. But where do we make NADH? The U-Hauls, everywhere. We got NADHs in glycolysis. We got NADHs in pyruvate oxidation. And we got NADHs in the citric acid cycle. That's why I call NADH the U-Haul truck, because there's more of them. All right, fumarate becomes malate. And then malate becomes oxaloacetate. But you're making a truck with this one too. You're making a loaded truck with that one too. So if you notice about this, catch it. This is a cycle. What you started out with is what you ended up with. Otherwise, it wouldn't be a cycle. Do you see that? I had a student who said, would you ever run out of oxaloacetate? No, because you use it, but then you remake it. Clever. It is very clever, yeah.
The succinyl-CoA goes into...
So what is it?
Succinyl?
This one?
Yeah.
Succinyl-CoA.
Succinyl. It goes into succinate.
Succinate? Yeah.
By releasing ATP?
No, again, remember what I was telling you? During this reaction, it loses a phosphate and gives it the ADP to make ATP. So I have a phosphate, I am succinyl-CoA, for example, and on my way of making succinate, I give the phosphate to ADP, and it becomes ATP. It's substrate level phosphorylation. The substrate gives the phosphate to ADP, and I become a product. The product is, in this case, succinate. So how do you remember all this stuff? Because I was a student in your chair long time ago, and I was up against this, and I'm like, oh my God, how are I remember this. 665 5 4s. Watch that. 665 and 1, 2, 3, 4, 5 4s. That's me remembering the carbons. Starting from citrate, 665 5 4s. Catching it? We're not done yet. I also remember this. Every time the number drops, that's a CO2 being released. Watch this. Every time the number drops, that's a CO2 being released. Every time I make a CO2, I make an NADH. What did I just say? I said, every time I release a CO2, I load a truck. I did not say it the other way. Do not say it the other way, because you will be wrong. If you said this, every time I load a truck, I make CO2. That's not true, because you do that here. Do you see a CO2? No. You also load 2 NADHs in glycolysis without CO2. You have to say it exactly like I'm saying it. Every time I release a CO2, I load a truck. You can't say it the other way. This is my way of remembering it. 665 and 5 4s. Every time the number drops, I'm releasing CO2. Every time I release a CO2, I'm making NADH. Are we okay so far? How about this one? This one is a weird one. Between the two sounds, I get ATP made by SLP. Call me weird. I don't care. I call myself, I remember it. You know, I can't tell you, I'm not going to give my age away maybe. I can't tell you the year that I took this stuff. You probably were not born, but I still remember it. Besides the fact that I teach it every semester, right? I remember it. It's called repetitive exposure, but it's also called strategy. I mean, I figure out that between the two sounding words, there's ATP made by substrate level phosphorylation. Now watch this one. Between succinate and watch. Fumarate, I make FADH2. See, I look for patterns. I look for, how do I remember it? Long term. Here's another one. Between fumarate and malate, I say boring. That means nothing going on. You catching it? Then all of a sudden, I remember my alphabetical order. L-M-N-O. L-M-N-O. Those are the things we do. Understanding it is one thing. Remembering it is a whole different ballgame. And you use those strategies. Yes, sir? Yeah. But a different truck. FADH2. Yeah.
So in between fumarate and malate,
nothing. Well, there is something, but nothing of your concern. So call it boring. Nothing going on. Well, something is happening, but you don't need to worry about it. So at the end of this, now watch this. This is one turn of the cycle. At the end of this, count them. You have 3 NADHs. You have 2 CO2s. You have 1 ATP and 1 FADH2. That's for one turn. But how many turns do we take? 2, because we have 2 acetyl-CoAs. We had 2 of these. So one of these will turn it once, the other one will turn it again. So now, what's the total number of NADH from the citric acid cycle? 6. What's the total number of CO2s from the citric acid cycle? 4. What about ATP? 2. FADH2? Yeah. Now look at the difference between you getting the question right on the exam. and God forbid, you're getting the question wrong on the exam. Watch this one. Listen to this one, I mean. I can ask you this question. Using one glucose molecule, how many ATPs do you get from the citric acid cycle? You get 2. Even though one turn makes 1 ATP, you actually gonna make 2 turns because from one glucose molecule, you make 2 of these. Now listen to the question, same exact question. I just remove one word, replace it with another. During the citric acid cycle, using one acetyl-CoA, how many ATPs do you make? 1. So if your eye does not fall on that word glucose and registers it as glucose, or if your eye does not fall on the word acetyl-CoA and registers it as, then you're going to get that question wrong. So what I'm trying to tell you is exam 3 is very tricky in the sense that you must read the question carefully. I mean, you can understand the material, you can memorize it. If you misread the question, you're going to go for the wrong answer because one of the choices is going to say 2 ATPs. Another choice is going to say 1 ATP, 6 ATPs, whatever. Read the questions carefully on, of course, on exam 2 on Monday, but also exam 3. Exam 3 is a monster because of that. And then you got photosynthesis in exam 2. You got photosystem 1, photosystem 2. If you don't read the 1 versus the 2, you're in a different completely area there. You're gonna get messed up. And then it also has mitosis and it also has meiosis. There's meiosis 1, there's meiosis 2. On the exam, you're gonna have chromosome, centrosome, centromere, centriole, chromatid, chromatin. Are you catching this here? All the words sound the same. What I'm asking you to do on exam 3, on all of your exams actually, but especially exam 3, read the questions carefully. The words look the same, but they mean completely different things. Centrosome is not the same as centromere that you're going to hear about later. Okay? So watch out, please, on this exam. So here's the final tally at the end of the citric acid cycle, and then we'll stop here, revisit the experiment, and we'll be done for the day. At the end of the citric acid cycle, you now have, this is from 1 glucose. From 1 glucose, you're going to have 2 ATPs from glycolysis. This is a net gain. You're going to have 2 NADHs from glycolysis. They go into the mitochondria, because remember, the meeting is in the mitochondria, so the NADHs go to the mitochondria. You're going to have 2 NADHs from pyruvate oxidation. They're already in the mitochondria. You have 2 CO2s, pyruvate oxidation. They leave your cell through simple diffusion because CO2 is small and non-charged. We talked about it. You have a total of 6 NADHs because we turn the cycle twice because you have 2 acetyl-CoAs. You have a total of 2 FADH2s from the citric acid cycle because again, you turn the cycle twice. Each one makes 1 times 2, 2. Same thing with this one. 2 ATPs from the citric acid cycle. 4 CO2s from the citric acid cycle. Now look at this. Do you remember glucose had 6 carbons? Do you remember that? Look what you're doing with the glucose that you paid so much money to buy and consume and eat and all that. You're taking all the 6 carbons and you're doing this with it. Breathing them out. Because that's what it is. Here are the 6 carbons of glucose. You're releasing them as waste. But what you're getting out of your glucose actually is the energy. That's what you're paying your money for. So when you go to the grocery store and you're buying food, know that a lot of it, you're going to simply go, release it as waste. But what you're actually paying for is the harnessed energy in this molecule. Because without it, you don't exist. So just to show you or give you a heads up of what we're going to talk about on Monday after the exam is this one. This is the oxidative phosphorylation. There are players in the complexes located in the inner membrane of the mitochondria. All your delivery trucks are right here now. Here are all of them all together. 2 NADHs from glycolysis. 2 NADHs from pyruvate oxidation. 6 NADHs from the citric acid cycle. 6 plus 2 plus 2 is 10. 2 FADH2s from the citric acid cycle. Here they are. They all gather in the meeting place, the mitochondrial matrix. And what are they about to do? They're about to do what delivery trucks do. And what do delivery trucks do? They deliver. What are they going to deliver? Don't say furniture. Electrons and hydrogen. Yeah. So they're going to deliver the electrons and hydrogen to the complexes. And that's when the fun really begins.